This is learning objective number 12 with Serena Singh, and in this video I'm going to be explaining how rearrangements in the genome can activate or inactivate genes. So first I'm going to give you a quick review of genomes in DNA. So the genome of a cell or an organism is the complete set of genes or genetic material present within that cell or organism. So DNA is the carrier of genetic information. Chromosomes are thread-like structures of nucleic acids and proteins which carry genetic information in the form of genes. Getting into our learning objective, we are going to now discuss rearrangements in the genome. So rearrangement in the genome refers to the movement of DNA within the genome. This results in an alteration of the location of this DNA in respect to the regulatory sequence of that genome. So the regulatory sequence of the genome includes the gene's promoter as well as an enhancer sequence. It's a segment of nucleic acid molecules which is capable of increasing or decreasing the expression of specific genes within an organism. When a regulatory sequence is bound by transcription factors, it is able to affect the transcription rate of genes within its vicinity. So this basically means that the rearrangement of DNA within a genome can have a great effect on whether or not a gene is active, aka expressed. This effect can be seen in the transformation of proto-oncogenes, which are genes that create proteins to regulate cell growth, into oncogenes, which are cancer-causing genes. The movement of DNA within a genome is just one way that proto-oncogenes become oncogenes. However, for the purposes of our video, we are only going to be focusing on the translocation of DNA as proto-oncogenes become oncogenes. So, cancer cells are frequently found to contain chromosomes that have translocated fragments. If a translocated proto-oncogene ends up near an especially active promoter, its transcription can increase, causing it to become an oncogene or cancer-causing gene. I have some handy diagrams here. Now, I know I've already described what translocation is, but it helps to have a physical representation of the process. So, as a quick refresher, translocation is the movement of DNA within the genome. So here we have two strands of genetic material. We are going to have a piece of this strand and a piece of this strand essentially switch places, so that after translocation occurs, each strand has a piece of the other. And when we do this, we see that there's really no net gain or loss of genetic information. This can be seen once again in this diagram where we have chromosome 11 and chromosome 5. Pieces of DNA from chromosome 11 are switched and placed onto chromosome 5 and vice versa. Now, in regards to our example of converting proto-oncogenes into oncogenes, we see that the movement of a proto-oncogene within the genome can cause it to be changed into an oncogene. If the new location of our proto-oncogene is in the vicinity of a regulatory sequence which contains a promoter, we have a resulting increase of transcription of that gene, and therefore activation of said gene. So what we're seeing here is the normal growth stimulating protein in excess. So this basically means that our proto-oncogene has become an oncogene because it is stimulating excessive growth of cells, which is cancer. So to finally sum up our learning objective, I am just going to say that rearrangement may activate or inactivate a gene. When a piece of DNA is moved within the genome, it can be found sometimes in a different location with respect to the regulatory sequence. Now, when it is in the vicinity of said regulatory sequence, we find an increase in transcription, which leads to the activation or inactivation of that gene.